So, good morning to all of you. Uh, Today, we will continue from where we have stopped in the last class. If my memory goes right, in the last class, we were discussing about the evolution of the life. And in this, we have tried to understand the primordial atmosphere on the earth, and we have also discussed how those parameters which prevailed on this early earth favor the origin of life. So today, uh, let's continue with that because till now we are not very definite about the primordial atmosphere. Whatever the things that we say is drawn from indirect evidences, and by that we have drawn the inference, and we are considering that primordial atmosphere must be like that, and this must have paved the way for the origin of life. uh, then the question is whether this can be scientifically validated or experimentally validated. Uh, till today, we fully agree to the uh, hypothesis that has been drawn by Haldane. So and Operin-Haldane hypothesis. This Operin-Haldane hypothesis, which suggested a model for the origin of life, was experimentally tested by two scientists, Urey and Miller, in 1953. Because unless we have any scientific proof or experimental evidences, it is very hard to believe only the hypothesis. Though logically the hypothesis may appear right, but we are not absolutely sure about the uh, truthfulness of that hypothesis. So for that, an experimental validation came from Urey and Miller in 1953. What they did was they tested the Operon-Haldane hypothesis by artificially creating an environment which might have been prevailed on the earth something 4 billion years ago. So this is and we know that, and we assume that during that period the atmosphere above the warmed sea water was containing uh, water vapor, hydrogen, methane, and ammonia. And there were lot of lightning, and which might have provided the energy for the origin of life. If you analyze the seawater also, it was full of weak chemicals like carbonic acid, hydrochloride, sulfuric acid, and all. So then this Miller and Urey thought of creating such an environment and find out whether the life can originate from this kind of uh, condition. So for that, they set up an experiment, and that is popularly known as Urey-Miller experiment. What they did was they took some glass vessels, and the glass vessels were joined as it is shown in the photograph. So, and inside which the water was containing uh, those chemicals, that is carbon dioxide, methane, hydrogen cyanide, and such things. Then, in order to create the energy, or in order to provide the energy, because we claim that during that period the energy mostly came from the lightning. So the here in order to supply the energy, the high voltage electrodes were fitted, and then what was done was uh, the water was boiled, and it was water vapor was created, and this water vapor was subjected to the electrical currents produced by the electrodes, especially in this chamber where the electrodes were discharging the electricity. It contained ammonia, hydrogen, and methane, and then later it was cooled, and this cooled water was collected and tested for chemical analysis. When Urey and Miller analyzed this water after a week of this experiment, they could find out 15 amino acids Basically, they have never put any amino acid there, but what they put were only methane, hydrogen, and ammonia, and by that they could produce 15 different kinds of amino acids. And all of you know that the amino acids are biologically very important molecules because they are the uh, base of for any protein. And even along with that, they got some of the biologically important molecules like ethanoic acid, lactic acid, and urea. Then later, the similar experiments were done using the carbon dioxide also. When the carbon dioxide was used in the second time, even they could get the nucleotides. And all being the student of biology, all of you know the nucleotides are the basis for this nucleic acid. That's RNA and DNA. When they are used along with along with ammonia, water vapor, and methane, they have also added carbon dioxide, and they could get the nucleotides. So, by this experiment, Urey and Miller could collect 15 different types of amino acids, ethanoic acid, lactic acid, urea, and when they have used carbon dioxide also, they got the nucleotides. So, by this, Urey and Miller came to certain conclusion. But one thing we should agree to the fact that though we have done the experiment in the laboratory, it cannot reproduce the exact condition of the primitive Earth, because the knowledge about the primitive Earth itself is very scarce. So that's why we should agree to the fact that whatever the experiments that we design in the laboratory, we cannot reproduce the exact condition of the primitive Earth with prevail. And even we don't know what exactly happened during this time. But certainly it can support what the this Operin and Halden has proposed. Basic building blocks for large macromolecules can be synthesized in vitro, that is in the laboratory, from inorganic components. All of you know the building blocks of larger macromolecules are organic one, but what we have used is mostly inorganic one. So that's why it is possible to synthesize organic large organic molecules in the laboratory from the inorganic compound. This is what the Urey-Miller experiment concludes. So these organic molecules were collected, and this supports what uh, Harden and Operin has proposed. That is the hot dilute soup concept what we have discussed. Now, what we got is the monomers. Initially, of course, even on the Earth, primitive Earth, what we might have or what might have been formed must have been the monomers of the polymers. That's the basic units of these polymers. And all of you know amino acids are the basic unit for protein. These amino acids, uh, they develop polypeptides. Polypeptide is a peptide bond. By developing the peptide bond, the amino acids develop a polypeptide bonds, especially in dry and highly concentrated monomers were heated. If you take the amino acids and in a dry and highly concentrated uh, medium, you heat it, you get polypeptides. The condensation reaction must have taken place, forming the peptide bonds between the amino acids, or there might have been phosphodiester bond between the nucleotides so formed. So two types of bonds were formed. One is the peptide bond between the amino acids, and another one is the phosphodiester bonds uh, between the different types of nucleotides. That is adenine, guanine, thymine, cytosine, all those things. So now, how this must have been formed? There must have been some catalyst. All of you know catalysts are the one which accelerate the reaction. A molecule, the clay minerals might have functioned as catalyst. That's what we think now. A molecule absorbs to the clay mineral particle, and when these molecules, specially attached to this particular condensation reaction. And these clay particles, what is known as conservates, may have been essential role in the catalysis and in the formation of the polymers. From monomers to polymers, the bond formation, either it may be the peptide bond or phosphodiester bond between the nucleotides, must have been achieved by the clay particles, because the clay particles might have performed the role of catalyst. That's what the assumption. So now, once polynucleotides are formed by the formation of phosphodiester bond, these polynucleotides show the tendency to copy themselves using the complementary base pairing. If you see the RNAs and RNA and DNA, there's the nucleic acids. There's the ribonucleic acid or deoxyribonucleic acid. Uh, they have the tendency to replicate themselves and using their complementary base pairing. Um, Again, this was probably catalyzed by the presence of clay particles and other metal ions, which prevailed during those times. And as a result, a single-stranded polynucleotide would have been formed, which can be equivalent to the RNA what we see today. So the nucleotides develop phosphodiester bonding between themselves. Because then once they start bonding, they are called as polynucleotides. And these polynucleotides, even today, they show the tendency of copying themselves or replicating themselves or reproducing themselves or multiplying themselves. And this particular process of replication was again catalyzed by the presence of clay particles, and lot of metal ions were also there in the medium. They might have catalyzed the reaction, and thus they formed a single stranded polynucleotide which has the capacity to replicate, and this must be equivalent to the present day RNA what we see today. So that's why it's not wrong if I say RNA was the probably the first hereditary molecule which is having the power to copy itself. It can replicate, because even today we know that RNA replicates or multiplication. This RNA, probably so-called RNA or RNA like polynucleotide, I should say, or probably the first hereditary molecule having the capacity to copy itself. And even RNA show enzymatic properties because it has ribozymes, and these polynucleotides were the good molecules because they could store and transmit the information. They are good molecules only because they store and transmit the information. Um, but you cannot compare that with the cell, because they are not versatile as the cell, because cell will carry out lot of other biochemical activities but not the RNAs. So these polynucleotides were good molecules because they can store and transmit the information when they replicate, but at the same time they are not as versatile as the cell, because cell will carry out lot many function, biochemical functions. So that's why this must be a very very prototype of life, only having the capacity to replicate, and at the same time not only replicate, uh, store and transmit the information from first copy to second copies. So then at the same time, amino acids were also formed. This amino acids joined by means of polypeptide peptide bonding. Um, and once the peptide bonding, a long polypeptide chain was formed, and which form complex three-dimensional structure. Uh, Usually, even today, we have the proteins exhibit primary, secondary, and tertiary structures because of the foldings, and which can form complex three-dimensional structure, and are much better at complex cell function. So they have role in the functioning of a cell. So what has happened? On one hand, polynucleotides were formed. On the other hand, polypeptides were also formed. Now, polypeptides are there, polynucleotides are there. Now, the only thing is there must be a partnership between polynucleotides and polypeptides. And then, in due course, a partnership must have developed uh, between these polynucleotides and polypeptides. And we don't know how exactly this might have happened, but probably when these polynucleotides and polypeptides they remain in the same environment and in the same vicinity, then somehow they might have come together and thought of making a partnership where polynucleotides and polypeptides both of them can contribute. So here what has happened was polynucleotides, as you know, they can direct the uh, they can replicate. But here, this polynucleotides, when it was amalgamated with the polypeptides, these polynucleotides started directing the synthesis of polypeptides. Uh, Later, polypeptide synthesis was under the information given by the polynucleotide. So this we can say that it's a clear path. What we can see a clear path of the travel of information or flow of information from polynucleotides to polypeptides. And this is what today we call it as translation. So the translation was started at that time. The clear information where the flow of information from polynucleotides to polypeptides. Even today we know that in the synthesis of the nucleic acid, translation is the first step where uh, the polypeptides are synthesized based on the messenger RNA information. So here also in initially these nucleotides started directing the synthesis of polypeptide because of the partnership. So this we can call it as the first hereditary material, and this is what we call it as RNA. And today we know that genetic information is usually stored in our DNA, but still in some organisms such as viruses use RNA to store this information. Whatever the RNA virus that we see, there only we have only RNA is the uh, genetic material which store the information. Even in this COVID-19, it's an RNA virus. So now, okay, now RNAs have been formed, and the polypeptides were formed as per the information given by the polynucleotides. What was there? There were short polymers of ribonucleotides. We can call it as now RNA monomers. Uh, and even today we can synthesize this in abiotically in the laboratory. RNA monomers even we can synthesize abiotically in the laboratory. And so even if in the laboratory, if these polymers that is are added to a solution of ribonucleotide monomers, a sequence up to 10 base long are copied from the template according to base pair rules. That is in the laboratory. If zinc is added, the copied sequence may reach even up to 40 nucleotides with less than 1% error. The copying with less than 1% error, and this is what the formation of short from RNA polymers, um, monomers to formation of short RNA polymers up to 10 base pairs. Then if you add zinc, then it will copy even up to 40 nucleotides with 1% error. This can be proved in the laboratory. It was in 1980s. One scientist by name Thomas Sach, he discovered that RNA molecules are important catalysts in modern cells. RNAs is the enzyme. In the modern cell, RNA molecules function as an important catalyst also. RNA catalysts are called as ribozymes. So, ribosomes also help in catalyzing the synthesis of new RNA polymers. So, the RNA formed, one thing, they started storing and copying the information. At the same time, many of these RNAs, what we call ribozymes, they themselves started functioning as the catalyst, which helped in the synthesis of new RNA polymers. Uh, Especially in the prebiotic world, RNA molecules must have been fully capable of ribosome catalyzed replication. So the ribozymes helped in the catalyzing the replication process. So these initially formed RNAs have performed two things. One is storing and passing the information from one generation to another generation by the process of replication. Second thing is, for this replication, the same RNAs in the name of ribozymes started catalyzing the reaction and accelerating the reaction, thus enhancing the speed of the reaction. So, and it was later in 1980s discovered by Thomas Sech, because the RNA molecule itself can catalyze the model cells. So this is RNA can fold up and function as an enzyme. This is the 3D model of an RNA clover leaf, and I'm not going to the details. When you study RNA, you can study them. So even the laboratory experiments suggested us that RNA sequence can evolve a biotic condition without any life, prebiotic condition or an abiotic condition, in the within the laboratory using the inorganic precursor, we can synthesize the RNA. And RNA molecules uh, have both genotype, that the nucleotide sequence which store the information, pass the information, and another one is they are also having the phenotype, that is the external appearance of a three-dimensional shape, which helped in interacting with the surrounding molecules. RNA molecules, both genotype, they can function as a genetic material, and they are also having a three-dimensional shape, and because of this three-dimensional shape, they can interact with the surrounding molecules. Under certain particular condition of the prebiotic earth, some RNA sequences are more stable, and this more stable RNA sequences replicated faster and with very less number of errors than compared to other ones. Because when the replication takes place, in order to pass on the information perfectly to the next one, the error must be less. So this copying error, some RNA sequences which are more stable, uh, which replicated faster, not only replicated faster with less number of errors, and then occasionally these copying errors were there, and this might have created the mutation, what you call point mutation or gene mutation. And out of these mutants, uh, there must be a screening mechanism where mutation for the most stable and best was selected and rest was eliminated. So when there is a mutation, if the mutation is beneficial, it was selected, the one which is most stable and best replicating was selected, and others were rejected. Then this RNAs directed the protein synthesis, as I have told you, when there is an amalgamation of RNA with the polypeptides. In the very beginning, as a weak binding of specific amino acids to the base of RNA molecules. This uh, friendship between the RNA and polypeptides, in the very beginning, it was a weak binding of certain amino acids to the basis of RNA, the nitrogen base of RNA molecule. Uh, then, as a result, this RNA base must have functioned as a template which can hold few amino acids together, which can support a few amino acids together long enough for them to be linked. So even today also, one of the important function of ribosomal RNA is the same thing. In the ribosomes, what they is the bases, nitrogen base of the RNA, they hold or support the amino acids to come together and form a chain. So this RNA polypeptide friendship led to the synthesis of a short polypeptide. By that, they started synthesizing short polypeptides, and these polypeptides behave as an enzyme, helping the RNA replication. Now here, amino acids are the basic unit of protein. What is protein? A long chain amino acid where large number of amino acids held together by a peptide bond. We call it as polypeptide chain. It is nothing but a protein. And you also know that all enzymes are protein. All enzymes are protein. So these short polypeptides, or in other words, we can say that small proteins with very low molecular weight because of short, only few bases are there. These short polypeptides, along with RNA, must have started behaving like an enzyme and helped RNA in replication. So that's why it was not wrong if I say it is a molecular cooperation. It is a molecular cooperation between these two things. Now, the okay, this is I will sorry, simple. Okay, now after this, uh, what might have happened was slowly now RNA is there and polypeptides are also there. This RNA, along with these polypeptides or proteins, they formed a cover around the RNA. The polypeptides formed a cover above this RNA. Now, RNA can synthesize the polypeptides of its desire, and these polypeptides can help in the replication process of the RNA by being enzymes. So, a symbiotic association was created between the two chemical molecules. So, that's why it is a molecular cooperation was established. And then, when this is a RNA code and surrounded by a protein. It's a very unique step, and we can call this as the protobionts. Protobionts, and they are very similar to that of the present day virus. Even if you have studied the virus, you know what is a virus. Inside there is an RNA code. Surrounding that there is a protein code. So now also these protobionts started showing the same feature, having RNA code and a protein code. And then the natural selection must have accelerated the protobionts to grow and replicate. And that would give rise to all the different organisms that we see today. So this is what we see. Once the primitive RNA genes and their polypeptide products were packed within a membrane, the protobionts could have evolved as units. What has happened now? RNA genes surrounded by that packed in a polypeptide, and all of them are sorted in a some what you call a membrane. This uh, started functioning as a unit to ensure. Okay. Huh? So now uh, the molecular cooperation started uh, because of the favorable components were concentrated together in the surrounding, and this led to a molecular cooperation. So now, how do we agree what just now I have suggested? There is a formation of a protobiont, or which can be similar to that of the present day virus. This is the photograph shows something 4.5 billion years old rock. It is meteorite rock, and once it must have been the part of the Mars and all those things. And today it is preserved in this one, and that on the surface you can expect this must be the protobionts of that time. Meteorites was destroyed. So this is a paleontological evidences on this. When they analyzed, they could see on the surface these were made out of RNA core and a protein cover. When they analyzed it, it was found. In 1980, Louis Pasteur conducted one experiment. All of you know Louis Pasteur, a well-known scientist even today. He tested whether the microorganisms, whether they emerge spontaneously. Spontaneous generation of microorganism, is it possible or not by reproduction of the existing microorganisms? And he conducted grew open containers, sterilized both, and then he suggested that microorganisms cannot be generated spontaneously. That is, without life, you cannot generate the life. Life originates from pre-existing life. That's what he has suggested. Now, we will keep this Pasteur hypothesis later. But now we will also think about one more hypothesis, that the iron sulfur world hypothesis. All this protobiont creation, where it must have taken place. So, as far as the new study says that this synthesis of the organic molecules formed at the hydrothermal vents in the ocean floor, because uh, this can be supported with the modern laboratory simulation studies. The catalytic effect of iron and nickel sulfide in the hydrothermal vents, because this iron and nickel sulfide can function as a catalyst. So, based on this, it is now agreed that this first protobiont must have been formed or created in the hydrothermal vent in the ocean floor, where there is a lot of iron and sulfur. And this iron and sulfur, nickel sulfide especially, favor the formation of so-called protobiont. Now, protobionts, we cannot call it as cell. Cell has totally different thing, because lot of biochemical activities are there within the cell. But in a protobiont, it can replicate, it can store the information, and protein can be from different types of amino acids can be synthesized as per the instruction given by the RNA. But to become a cell, it has to perform much more than what a protobiont can do. Then the question comes, how the cell must was formed. Spontaneous assembly, the only one word which we can say that, spontaneous assembly of small organic molecules into macromolecules, because cell is a macromolecule, and spontaneous assembly of these protobionts molecules, and as a result the protobionts evolved. It might have taken long time duration, and these protobionts evolved similar to the cells. What are the capacity? What is the nature of the cell? it should be able to divide, it should be able to maintain homeostasis, that the constant internal environment, and there must be catalytic activity for the biochemical synthesis. So the protobions must have attained and achieved these characters, and they become similar to the cells. So see here the microspheres, a type of protobions, they might have came together and uh, assembly of a new cell was formed here. So here, first in this particular process of the formation of the macromolecule, microspheres were formed, and these microspheres were formed in the protobions from water and polypeptides, and slowly they have developed an electrical gradient on the surface, and these microspheres were having a outside membrane which was selectively permeable, as the as all of you know, the present day plasma membranes are also having the same characteristic feature of selective permeability. So these microspheres from the protobions were formed from water and polypeptide by developing an electrical gradient on the surface. By developing an electrical gradient on the surface, they become selectively permeable, and this selective permeability is the characteristic feature of all plasma membranes. The Now, in the first step in the evolution was from RNA to cell to DNA and protein synthesis. It's an important step. This is started with RNA. All of us know that evolution is a directed one or directed evolution. Now, in nature, what was available was large pool of RNA molecules with different sequences. As a result, nature has got a chance to select the desirable RNA molecule with different sequences and to catalyze them. How? They can amplify, mutate, and repeat. By this process of selecting the required RNA molecules with different sequences, and by having the ability to catalyze the reaction, say to amplify the sequences, mutate the sequence, or repeat the sequences, uh, evolution has started. See, this is the directed evolution where you can see large pool of RNA molecules and selection for the ability to catalyze the chemical reaction. Then what has happened? Some molecules with some ability to catalyze were selected, and where you can amplify them and mutate them to create another large pool of similar RNA molecules. By amplification and mutation, what you can do is you can create a large pool of similar RNA molecule. Even you can repeat the selection and amplification and mutation process. As a result, what we got is molecules with best ability to catalyze the reaction. So now it is unidirectional. So this early catalysis is a very important step in the origin of life, because as the molecules absorb the clay mineral particle, they become concentrated. These clay particles, as I have already told you, what is known as conservates, they have become very essential for catalysts in the formation of polymers. So again, to repeat what we have discussed, once polynucleotides show the tendency to copy themselves and they were catalyzed and equivalent to RNA were formed, and RNAs we discussing And polynucleotides were very good molecules in storing and transmitting the information, but they were not versatility. A great partnership has happened between polypeptides and the RNAs, which can be called translation. Later, the hereditary information was probably stored in the form of DNA because DNA is more stable than RNA. Initially, it was RNA, but later it was stored in the DNA as DNA is more stable. Even today, passage of information from RNA to DNA is possible, where you can see the reverse transcriptase enzyme of retroviruses show that because the viruses use the host DNA to produce their own RNA. So by reverse transcriptase, you know that from RNA, the information can pass from RNA to DNA, and as a result, the DNA must have origin. If a piece of RNA codes for a particular good protein, uh, then protein being used for RNA molecules. But here, what is the importance is the development of the first membrane. Suppose if the RNA is enclosed in a membrane, where it can keep its protein to itself and gain the selective advantage. So, in course of evolution, to get the selective advantage of being kept with this protein without sharing, the membranes must have evolved. So, the membrane probably pushed the evolution by natural selection by forward. Simultaneously, when the first cell was formed, till now no oxygen was there, no free oxygen. All these things are chemosynthesizing. It is said that cyanobacteria first evolved with photosynthetic activity because the cyanobacteria has the ability to split water into hydrogen and water, and that release the oxygen. Till now, all metabolism was anaerobic in the absence of oxygen. So only after the cyanobacteria, which got the capacity to split water into hydrogen and oxygen, oxygen in the atmosphere made possible the evolution of anaerobic metabolism. And only during this time, the ozone layer started developing, only when free oxygen was available. How do we find that? What with cyanobacteria are the first one. Even we today we see this living bacterial mass and even the fossilized bacterial mass, which dates back to billions of years old, suggest that the cyanobacteria was the first bacteria to evolve and having the capacity for so-called photosynthesis. They are known as stromatolites. Stromatolites. They are all the uh, patches of this bacteria. And some of the oldest fossils are something 3.5 billion years old, which you can see, the cyanobacteria. So then, till now we have seen how the life evolved from non-life. Now the question is life evolving from non-life even today? In order to find an answer for this question, especially Louis Pasteur and others conducted a lot of experiments, and they were convinced that life does not come from non-life on earth, today, because only in the atmosphere or the condition which prevailed during that time favored the origin of life from non-life. But the present day conditions are not favorable for the origin of new life from non-life. So that's the one. And bacteria, which is already existing today, would eat any sort of prebiotic soup, and it is not possible for the origin of new life from non-life. So this is what I wanted to share with you today, and I and this is how the life originated, first life. And once the life originates means then evolution of life is totally different thing, which we will see in the next class. So is it clear? Can you hear me, Sudha?
Yes, sir.
Okay. All the slides were visible to you.
Yes, sir.
Okay. Any questions or anything which you have not followed? So about this write up, probably tonight I will give you send a write up. I have a write up, but only in the evening I will be able to share that with you through WhatsApp. Anything else? Any questions? Or tell me if you have not followed anything. If you want me to repeat anything. If not, we will close here and we will meet in the next class. Is it okay?
Okay, sir.
Okay, thank you.
Thank you, sir.
Thank you, sir.
Okay, it's my pleasure. Okay, we will meet in the next class.